Good afternoon to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, let's begin the session. Let's begin the session wherein first before starting the session, today we are going to deal with plant physiology diagram based questions itself. So I am going to firstly say you about or introduce the courses like NEET World special programs, where you have 3 programs. One for 11th standard students, you land on NEET World's 2-step to medicine with the minimal fee of 3333 per month. Second is NEET World special program, that is 1-step to medicine for 12th standard students with the fee of again 3333 per month, and NEET World's power step to medicine, wherein the fee is again 3333. For any program the fee is the same per month. Okay. So yes, this is highly useful for all the students of any financial background. So the classes will be run both in the offline and online mode with complete study materials, and the program is designed as such with 2 rapid revision session programs. Okay. And to know more about this programs, you can call on to the given number, or you can directly visit our branch which is in Narayanguda, Hyderabad. So let's get started with the questionnaire. First, look at this question. Consider the above given figure and select the options that can be best concluded from it. So to solve this question, you should have a clear idea on what is action spectra and what is absorption spectra. Absorption spectra and action spectra, both of them are given by which scientist ma? E.W. Engelmann. Okay, both of them are given by E.W. Engelmann. He has proposed both absorption spectra and action spectra. What is absorption spectra? Different pigments absorbing sunlight at different wavelengths is called as absorption spectra. If that is placed in a graph, that is called as absorption spectra. What is action spectra? Maximum wavelength of light absorbed by one pigment, and that conducts maximum amount of photosynthesis and maximum oxygen evolution. That is what? Action spectra. So here if you see beta, consider this diagram and what does this actually say? This is showing you that chlorophyll A, when in absorption spectra you have observed both the graphs. In absorption spectra you have plotted different pigments at different wavelengths like chlorophyll A, chlorophyll B, carotenoids and xanthophylls, where the diagram is very clear in your NCERT. In action spectra if you see, from absorption spectra you can clearly predict that chlorophyll A is the pigment which is absorbing maximum wavelength of light. So in action spectra chlorophyll A is only predicted. In this graph you can see chlorophyll A of absorption spectra will coincide with that of the action spectra. Okay, look into the options once. The action spectra shows a graphic representation of amount of light of different wavelengths absorbed by a pigment. It is not about different wavelengths. Absorption spectra depicts the relative rates of photosynthesis at different wavelengths. See, this is not absorption spectra, this is action spectra. So here relative rates of photosynthesis at different wavelengths of light, this is not predicted. Action spectra is corresponding to the absorption spectra of chlorophyll A here. So here action spectra is corresponding to the absorption spectra of chlorophyll A, because chlorophyll A will absorb maximum amount of light and it will conduct maximum photosynthesis and maximum oxygen evolution. So here what will be the right answer beta? Option C will be the right answer. This is not about this is action spectra, till here it is correct only, but this is not showing different wavelengths of the light. This is not at all the absorption spectra, this is what? Action spectra. So option B will be wrong. C is correct because this is clearly predicting what? action spectra is corresponding with the absorption spectra of chlorophyll A. Clear ma? Next question you see. This is the simplest question from just the chloroplast diagram. You see, X is what? The stalk of sac-like structures. The stalk of sac-like structures are called as what? Grana. So, grana are the sites or locations of what? Light reaction. So, you can plot X as light reactions. Okay. Y, which is a fluid filled space in Y. In this fluid filled space, what will happen? Carbon dioxide fixation or carbohydrates or glucose synthesis will happen. That synthesis is taking place in the fluid filled space, that is stroma. So, here what is happening? Carbohydrate synthesis, and these granules are what? Starch granules, where the glucose is stored in the form of starch inside the chloroplast. Okay. Glucose, to store it will get stored in the form of starch. Here one point I would like to convey you. Glucose, if it has to store, it will store in the form of starch. Glucose, if it has to transport, for transport or translocation, it will be transported in the form of sucrose. Fine, it will be transported in the form of sucrose. To store, it will be stored in the form of starch inside starch granules in the chloroplast stroma. To translocate or transport, it will be converted in the form of what? Sucrose. So, here what will be your option beta? Z is what? Starch granule. So, it is carbohydrate storage. Our right option will be option B. Next question you look into this one. Given figure depicts the light harvesting complex of photosystem 1. So, this is about PS1. When it is PS1, the reaction center will be what? P700, right? The reaction center will be P700, and we know all these pigments or these molecules are what? Accessory or antenna pigments. Accessory or antenna pigments. Fine. So, A is what? Accessory or antenna pigments. Fine. Here B is what? This is the core molecule. B is the core molecule. Fine. And this is what? The reaction center, where you can say, as he has mentioned, it is PS1. The reaction center will be what beta? P700 will be the reaction center. And this is what? The primary electron acceptor. This is the primary electron acceptor. Fine. So, our option will be what ma? B is what? Antenna pigments, sorry. A is what? Antenna molecules or accessory molecules. B is what? Core molecule, and C will be P700, that is reaction center, and D will be what? Primary electron acceptor. So, here our answer will be option B. Fine ma? Understood, right? If here he has mentioned instead of PS1, he is mentioning PS2, then your answer will be the reaction center is at P680. Okay. If he has mentioned PS2, then the reaction center C will be what? P680. Remember that. Next, look at here, next question ma. Try to answer, online students. Study the given flowchart of cyclic photophosphorylation. So, he is asking you about cyclic photophosphorylation and select the correct option of A, B and C. So, here if you see ma, cyclic photophosphorylation, where will be this operated? This is operated or this is conducted inside the stroma lamellae. This is conducted inside what? Stroma lamellae, wherein stroma lamellae will be having only what? PS1. Okay. And mostly cyclic photophosphorylation will happen in bacterial photosynthesis. Bacterial photosynthesis. This bacterial photosynthesis is also called as anoxygenic photosynthesis. Anoxygenic photosynthesis. Clear ma? Here there will not be any splitting of water or photolysis of water. There will not be any photolysis of water. Why there will not be any photolysis of water? Because of the absence of PS2. Clear ma? These are the important points, please take a note of it. So, look into this question once again. Of course, this A will be what? PS1 only, because only photosystem 1 will be present in the stroma lamellae, that only will conduct what? Cyclic photophosphorylation. So, here A will be what ma? PS1. A is PS1. B will be what? Electron acceptor. B is electron acceptor. See the reaction center. The reaction center, when it is cyclic photophosphorylation and in stroma lamellae only PS1 is present, then what will be C? C is P700. Then what will be the answer? B will be the correct answer. Next question you look into this one. Same type of question. Given below is a light harvesting complex of photosystem 1 and select the correct option he is saying. Of course, C is what? Antenna or accessory pigments. Antenna or accessory molecules. Fine. B will be what? Core pigment. This is what? Reaction center or the electron acceptor, you can say. This is the electron acceptor. So, here C is what beta? C is pigment molecules or antenna pigments, antenna accessory molecules or pigments, or pigment molecules is also one and the same. Pigment molecules. So, these are called as antenna pigments, accessory pigments or pigment molecules, and B is what? Reaction center or core pigment, and this is what? A is the electron acceptor. So, here what is the right option ma? Option 2 will be the right answer. Next question you see, this one. Chemiosmotic synthesis of ATP is shown in the diagram in thylakoid. Identify A, B, C and D. So, from the chemiosmotic hypothesis, first you have to know the labeling beta. Here it will be PS2, photosystem 2, fine, and this is what? Photosystem 1, PS1, and in between there is what? Cytochrome B6F. This is cytochrome B6F. This H plus ions will move to the lumen through what? Plastoquinone in the form of PQH2, where the electrons are given to, see, look at here, plastoquinone, when it takes 2 electrons, it will accept protons from the stroma, and it gets converted to PQH2, wherein protons are submitted to the lumen and electrons are being transmitted to what? Cytochrome B6F. From here again, you know, photosystem 1 will be there here at the end, and the ultimate electron acceptor is what? This is PS1. The ultimate electron acceptor is NADP plus. This accepting 2 electrons along with H plus in the stroma forms NADPH plus H plus. Fine. Once you look at it clearly, I will tell you the chemiosmotic hypothesis in a gist, that the proton gradient is generated from in between stroma and the thylakoid lumen. In between the stroma and thylakoid lumen, what is generated beta? Proton gradient is generated. How is that generated? Because the proton molecules will be decreasing in the stroma by 3 events. First event is through this ETS, the plastoquinone, when it gets the electrons, it parallelly accepts H plus from the stroma, and this H plus from the stroma are being sent to what? Thylakoid lumen. Second, in the beginning only, due to photolysis of water, water gets split into protons, electrons and oxygen atoms. These protons will get accumulated in the thylakoid lumen. Third, whichever minimal H plus is present in the stroma, that H plus are utilized by the ultimate electron acceptor of non-cyclic photophosphorylation, that is NADP plus. This NADP plus will accept the proton and it will be converted to what? NADPH plus H plus. So, because of these three events, protons will be decreasing or reduced in the stroma, and in lumen you can find more amount of H plus. So, here you see ma, A is what? Photosystem 2, C will be photosystem 1 and B will be cytochrome complex. D is what? ATP synthase. Through this ATP synthase proton channel, the H plus will be directed from lumen to stroma, which helps in the synthesis of ATP. So, here the right option will be option 2. Next question you see. Identify the reaction of photosynthesis and type of photosystem involved. What is the type of photosystem involved? Of course, this is what? Cyclic photophosphorylation, and this undoubtedly involves only photosystem 1. Just now we have seen in the previous objective, this is another model of it. So, here this is cyclic photophosphorylation, and how many photosystems are involved beta? Only one, PS1 is involved. This you see, identify the parts marked as A, B, C in the given figure, showing what? ATP synthesis through chemiosmosis. So, here if you see PS1 and here it is plastoquinone, cytochrome B6S, and here it is photosystem 2. So, you can see that clear event. The very first is what? Hydrolysis of water, okay, photolysis or hydrolysis of water. Second is what? Through plastoquinone, plastohydroquinone, where protons are pumped from stroma to lumen. Third event is what? The NADP plus will accept the protons in the stroma and it will convert or reduce to NADPH. Because of these three events, H plus will be reduced in the stroma and H plus concentration will increase in the lumen. This will generate the proton gradient, and finally ATP synthesis will happen by pumping back the protons from lumen to stroma through what? ATP synthase pump. So, this ATP synthase pump will have two channels ma, F0, F1 channels. F1 will be embedded inside the membrane, F0 will be projected outside. This is F1 and F0 will be projected outside. This is F0. So, here you can directly answer. First A is what? Stroma. Look into the options. See, A is here, A is what? Thylakoid lumen, and B here is what? F1, and C is what? This one which is projected towards the stroma, that is F0. So, here the option will be B. Remember, it is very simple beta. From ATP synthase, F1 will be embedded in the thylakoid membrane, F0 will be placed towards the stroma. Remember that. Okay, so, here A is what? Thylakoid lumen, B which is embedded inside the membrane, that is F1, and C this channel which is projected towards the stroma, this is what? F0. Fine. Next question. Here you see again the same question, but in a different pattern, asked about what? Chemiosmotic hypothesis. Here also you observe ATP synthesis is shown in the diagram in chloroplast of plant cell. Correctly identify A, B and C. A here is what? Thylakoid membrane. B here is what? Thylakoid lumen, and C is what? Stroma. So, here the option is 2. Very simple, right? Next. Identify all these labelings which are provided from what? Non-cyclic photophosphorylation. Non-cyclic photophosphorylation. Or you will also call it as what? Z scheme. Look at it carefully. First, this will be what? A is what? Photosystem 2. So, this is A will be photosystem 2, and Next you look into B will be what? Electron acceptor. This is electron acceptor, and then this is what? ETS, electron transport system. Here it is electron acceptor and this is PS1. Here it is PS2. Okay. And the last electron acceptor will be NADP plus, but the arrow is towards this. We have to identify E, means the one which is synthesized from NADP plus. We know after accepting two electrons, electrons being accepted by NADP plus along with H plus through the enzyme NADP reductase, it gets reduced to what? NADPH plus H plus. Clear ma? So, here A will be photosystem 2, then B is what? Electron acceptor, this is electron transport system, photosystem 1. Here the arrow is projected towards outer side. The ultimate electron acceptor is NADP plus. Accepting the electrons along combining with proton, it will be reduced to what? NADPH by the enzyme NADP reductase. So, here you look into it properly. What will be the options? PS2, electron acceptor, ETS, photosystem 1 and NADPH plus H plus. So, here the option will be 4. Clear ma? A will be photosystem 2. Yes, that is correct. And the right answer will be option 4 here. Next, choose the correct combinations of labeling the carbohydrate molecule involved in the Kelvin cycle. Okay. So, here you look into, this is 1, 2 and 3. So, carbon dioxide is first accepted by what in the Kelvin cycle? RuBp. So, 1 will be RuBp here, and this is fixed as 2 molecules of phosphoglyceric acid. So, 2 is what here beta? Phosphoglyceric acid, okay, or you can say triose phosphate or C, RuBp, and 2 is what? Phosphoglyceric acid. And 3 here you see this phosphoglyceric acid undergoing reduction and it will get reduced to glyceraldehyde 3-phosphate, where we will also call it as what? Triose phosphate. So, here the right option will be option 4. Clear ma? Next question. This you see, in the given representation of Kelvin cycle, identify A, B and C. Same type of question, where this will be what? RuBp, which is the primary carbon dioxide acceptor in C3 cycle, and this gets fixed to 2 molecules of phosphoglyceric acid, which is reduced to 2 molecules of glyceraldehyde 3-phosphate, which is also called as triose phosphate. So, here the answer will be again the same. Okay. Here it is option B. Next question. This is from C4 cycle. Okay. So, here you see answer from C4 cycle ma. Given figure represents C4 pathway. Select the suitable options for A, B and C. Here A is what? Fixation, carbon dioxide fixation or carboxylation, one and the same. So, here you can have a dot on all these 3 options. B is what? Decarboxylation, where C4 acid, be it malic acid or aspartic acid, this gets decarboxylated to pyruvic acid and carbon dioxide. So, here it is what? Decarboxylation. Fine. And next, C is what? Regeneration. The 3 carbon compound that is pyruvic acid is regenerated back to the mesophyll cells, whereas this carbon dioxide will be conducting Kelvin cycle. So, here first A is what? Fixation or carboxylation, B is decarboxylation and C is regeneration. Here the right answer will be option D. This you look into beta. Identify A, B, C and D in the given figure and you have to choose the correct option accordingly. A is what here? mesophyll cells. A is mesophyll cells and B is here what? Fixation or carboxylation. Here it is B, fixation or carboxylation. And C here is what, Nana? Bundle sheath cells. D here is what? C4 acid undergoing decarboxylation. So here you look into it properly. A is what? Mesophyll cells and B is fixation. Mesophyll cells, B is fixation, C bundle sheath cells and D will be decarboxylation. So the right answer will be option 1. Here you see the diagram, answer to this question also, ma. The diagram given below shows the relationship between light intensity and carbon dioxide concentration on rate of photosynthesis. So here light intensity affect carbon dioxide concentration and rate of photosynthesis are plotted. This is from factors affecting photosynthesis. Look into it properly, ma. Options correctly matched he is asking, which is correctly displaying about this graph. At higher light intensities, this is higher light intensities, gradually the rate does not show further increase as the light dependent reaction involving carbon dioxide becomes rate limiting. This becomes what? Rate limiting step. Option 2 you see, light is rarely a limiting factor, this is true, but we need that correct statement which is explaining this diagram about light intensity, carbon dioxide and rate of photosynthesis. Look into each option carefully. Photosynthesis is independent of the light, wrong. Increase in incident light beyond a point causes the breakdown of chlorophyll and a decrease in photosynthesis. So here what will be the right answer, ma? Right, light is rarely a limiting factor, this is true only, but this is not explaining the diagram. So we can't choose this. This you see, photosynthesis is independent of light intensity, wrong. So this we can't choose, that is a wrong statement. Option 1 you look into carefully. At higher light intensity, gradually the rate of photosynthesis does not show further increase as the light dependent reaction involves carbon dioxide becomes rate limiting, involving carbon dioxide becomes rate limiting. Look into 4. So here the right option with respect to carbon dioxide concentration, light intensity and photosynthesis will be 4 only, ma. Okay, because as you further increase, as light dependent reactions involve carbon dioxide becomes rate limiting. See, carbon dioxide concentration will mostly affect what? Light independent reactions. Okay, those are dark reactions C3, C4 CAM, because it shows its effect on the RUBP and PEP, which are the primary carbon dioxide fixers. So it is not about light dependent reactions. So we need not to choose option 1. So here the right answer will be option 4. Clear, ma? Next question. So this will be our last question about the factors affecting photosynthesis. I'd like to end this session on again saying you about the special programs of NEET World. 2 steps to medicine for 11th standard students who are aiming for NEET 2027 and 1 step to medicine for 12th standard students, power step to medicine for repeaters. All of the courses are in minimal affordable fee of 3333 per month. You can call us on the given number to get about the more updates about this program, or you can directly visit our branch. And this is the most crucial point in the public platform where all students can get benefited is to join our Telegram channel, that is NEET World's Telegram channel, wherein in this channel daily quizzes of biology, that is botany, zoology, chemistry, physics and mathematics will be updated. And second point is live classes schedules are given. Thirdly, free resources are provided like materials or whatever content is required for that day schedules are provided, and one-on-one doubt solving session with faculties are provided in this Telegram channel. To join, you can directly scan this QR code, or you can call to this number, or you can directly download through our website. Okay? So, this is all about today. Meet you tomorrow with a new chapter. Thank you.